The Lion hypothesis is the inactivation of an X chromosome, which occurs randomly in somatic cells during early embryonic development. Once the X chromosome is deactivated, all progeny cells will have the same X chromosome deactivated. This is done so that cells that have two X chromosomes, such as female humans, won't have twice the amount of protein production as organisms with one X chromosome, such as male humans. In order to inactivate the X chromosome, it becomes condensed and is pushed off to the side of the cell in order to prevent becoming expressed. This condensed X chromosome is called a Barr body. Early on in development, each cell randomly inactivates an X chromosome. Since some cells have one X inactivated and others have the other X inactivated, the cells may express different alleles from the X chromosome. This can be seen in female tortoiseshell cats. The fur color is an X-linked trait where big B produces black fur, little b produces yellow fur, and a female that's hetero presents both black and yellow fur called tortoiseshell. If I were to add in another trait for the cats where long hair is dominant and indicated by a big H, while short hair is recessive and denoted by a little h. If I crossed a yellow short-haired female with a black male heterozygous for long hair and was curious about the ratio of phenotypes in their progeny, I could utilize a branch diagram to figure it out. Since my first category is color, I do Punnett square where I'd write the mating male on top. Since he's black, he must be X big B and Y. The mating female on the side would be homozygous X little b. As seen, the females would be tortoise and the males would be yellow. I'll start by writing 0 black, half yellow, and half tortoise in my branch diagram. Next, I do the Punnett square for hair length. Since the mating male is heterozygous for long hair, he is big H and little h. The mating female is short-haired, making her little h, little h. It can then be seen that half would have long hair and half would have short hair. I can then add that to my branch diagram. Then to calculate my combined phenotypes, I'd multiply the fractions since I'm looking for a cat with both qualities. So the ratio would be 0 long black, 0 short black, 1/4 long yellow, 1/4 short yellow, 1/4 long tortoise, 1/4 short tortoise. If I loved how my tortoise-colored cat looked and wanted an exact clone of her, I'd be out of luck. Due to the random nature of inactivation of X chromosomes, each cat would have different cells in different places expressing this different alleles for hair color. Each cat would be its own unique mosaic of color.